As the plant body absorbs and accumulates various elements present in the soil and air, accurate monitoring of element kinetic is essential to understand their physiology. Accordingly, in the field of plant science, there is a significant need to investigate the physiological function responsible for absorbing, transferring, and accumulating the essential and harmful elements present in environments of varying temperature, humidity, light intensity, and atmospheric composition. Although radiography methods, despite a major limitation in terms of terminating the physiological activity of the test plants, have contributed immensely to the field of plant science. In recent years, various imaging technologies have been developed for investigating living systems. A very popular technique is radioisotope, also known as RRI imaging, which utilizes image of the absorption of the RRIs of a target elements from roots to leaf to monitor the movement and distributions within the plant body. So in this video, we present an outline of RRI imaging technology in plants with a special focus on investigating element kinetics. RRI imaging systems employed in plant science research are based either on the introduction of radio tracers or on the improvement of existing medical or preclinical RRI imaging system. However, in contrast to the medical field, the understanding of element kinetic is the main concern in plant science research. For example, in nuclear medicine, positron emission tomography, also known as PET, and single photon emission computed tomography, also known as SPECT, SPECT, involve radio tracers designed to congregate at specific sites. This implies that the imaging apparatus must quickly identify the point where the positron emitting nuclide aggregate. On the contrary, in vivo tracking of the movement of various elements is of interest in plant science, and therefore multiple images of the corresponding radioisotope need to be simultaneously monitored. Most of the medical or preclinical RRI imaging systems acquire image using a limited variety of RRI, such as single gamma emitting radionuclide, positron emitting radionuclide, and etc. Furthermore, the radiation signal from RRIs vary between beta rays and X-rays and gamma rays due to their energy difference. Therefore, the investigation in plant science requires an imaging system capable of covering a wide variety of elements. This is the central focus in the development of plant RRI imaging technologies. In recent years, there has been a dramatic evolution of fundamental technologies for radiation detectors such as scintillator materials, semiconductor detectors, and electronic circuits, leading to a high degree of freedom in their design. For example, silicon photomultiplier SIPMs, which are the advancement of photomultiplier tube and are widely used compared to PMTs. SIPMs have several advantages, such as utilization of a small operating voltage, high durability, a smaller size, lighter weight, and higher magnetic field resistance. However, SIPMs sometimes require the development of application-specific integrated circuit, which could be a cost prohibitive factor. The development of ASICs has recently become more straightforward, and several novel RRI imaging systems with integrated SIPM have been developed. Depending on the task involved in target plant physiologies, a specific plant RI imaging device with a measurement principle best suited for radiation detection should be used for studying element kinetics. In other words, for plant imaging experiment, multiple highly versatile technology are required rather than one state-of-the-art technology for a specific application. Radioisotope imaging has developed varieties of technology for specific detection in plant. For example, real-time radioisotope imaging system, also known as RRIS, positron imaging technology, or positron emitting tracer imaging system (PETIS), gamma camera imaging technology, and prospective RI imaging technologies. Real-time radioisotope is used to detect X-rays and beta rays via plant leaf and roots. RRIS has 1,000 times better range of detection compared to the traditional system because it can visualize beta rays in thick sample. Besides, positron imaging can estimate the position of the desired chemical element by utilizing the positron emitting radionuclide's physical characteristic. The positron imaging technology has sensitivity at higher degree than other techniques of radionuclide-based imaging. For instance, by using carbon-13, which labeled as carbon dioxide, can detect the kinetic image of fast elements, such as sucrose, which can enhance our understanding on the plant photosynthesis and productivity. Last but not least, gamma camera imaging can be used for agriculture research by visualizing desired element kinetic in few hour unit or days with long half-life. Radioisotope imaging. For example, gamma camera imaging has been used in physiological functions such as in utilizing high level of cesium in plants for remediation of soil and discovering the radioactive cesium accumulation in food crops caused by the absorption and transfer of radioactive substance in the food. In conclusion, radioisotope imaging has a wide horizon in plant science and agriculture application and can contribute an enhancement in research area. For example, the imaging of water, gas, and other elements can be detected by using radioisotope imaging. Then, some of the researchers are interested to contribute in this field by discovering a prospective imaging system technique. Compton cameras has been developed and gained attention as the future radioisotope imaging technique, which is non-invasive because it can overcome the usable limitation of positron imaging and gamma camera, which has low sensitivity. It can precisely measure scattering of content by the detector and obtain the radioactive isotope distribution resulting from the estimation of the gamma rays arrivals direction. The discovery can also allow to analyze the kinetics of multiple elements in a sample body of plants simultaneously. For instance, this radioisotope imaging device has been developed by researchers in order to environmental monitoring of cesium for the post-disaster of Fukushima. Therefore, the Compton camera is required for the detector elements development, which has high energy resolution as well as electronic circuits that can bear measurement data at enormous amounts.